Okay, thank you so much. I'm really glad to be here and to give this presentation, and I would like to thank the organizers for giving me the opportunity to speak about our company. We are a fairly new company. We exist basically about a year, and we have offices both in Israel, in the UK, at the Cambridge University area, and in Boston. And despite existing only a year, we set up quite interesting collaborations and connections. So first, as I said, we are operating within the Cambridge University. We have a startup agreement, so our R&D lab is within the university with access to all their facility, which allowed us really to move very fast from ideation to execution. We also have a CRADA agreement, a co-development agreement with the Vaccine Research Center at the NIAID. And I'm going to describe one project that we're doing together about SARS-CoV-2. We were quite lucky to receive funding from the Gates Foundation that funded some of the projects that I'm going to present today. And we have a lot of collaborations around technology development with Twist, and I'm going to highlight those collaborations as well. So basically, my talk has two parts. In the first part, I'm going to tell you how we are creating an RNAi prophylaxis against SARS-CoV-2, and this is in collaboration with NIAID. And in my second part, I'm going to tell you how we created a replicon system for SARS-CoV-2 that really allows us to safely simulate this virus in an environment that is not BSL-3, and we think it can really accelerate R&D efforts. And this is something we are particularly excited about. We got the results just in the last few days, and I added them to this presentation. And this is a project that we are doing with our collaborators at Twist. So let's move to the first part of this talk about the RNAi prophylaxis. And maybe one question that you have is, why the hell we need yet another prophylaxis for SARS-CoV-2? And to answer this question, I would like to describe the situation in Israel. Israel was one of the first countries in the world to get the vaccines and to start a population-wide effort. So already back in end of January, we vaccinated most of the population. And these were like amazing days, right? The cases were going down. We opened up many things. I went to these large concerts with my wife. It was beautiful. But then what really happened is, and we vaccinated quite a lot of the population, as you can see over here. But if you look at the vaccine effectiveness, and this is raw data that I got from the public dashboard of the Ministry of Health, you can see that kind of like in around April, people who got the second shot were quite protected with over like about 93% efficacy of the vaccine to prevent cases. But then as the summer started, we started to see a very rapid decline in the effectiveness of the vaccines, and we used mainly the Pfizer vaccine. So these vaccines are waning quite quickly. They're amazing. They're doing a great job, especially in preventing severe diseases, but they can wane quite quickly. And what happened is that during the summer, from June, we were one of the safest countries in the world with very low number, less than 0.1 per, or less than 1 per million COVID cases. And within 10 weeks, we got to a situation where we had about 100 cases per 1,000 individuals, more than the US at that time. Now the situation is better because we boosted people, but the question is, can we just keep boosting individuals, and how frequently should give these boosts? And why not to design more types of prophylaxis that aim to reduce transmission specifically? So we envision a new type of prophylaxis that is based on nasal spray that delivers an siRNA cocktail against SARS-CoV-2. Now this cocktail will enter into your epithelial cells in the nose and will convert this very nice environment for the virus to replicate into a hostile environment. So as such, it is totally orthogonal to your immune system, and therefore it can work. It can add another layer above the vaccines. It has no sensitivity to spike mutations because we don't need to target the spike protein. We can target other parts of the genome, including non-coding regions, or also regions that are just not in the envelope of the virus. So the immune system have harder time to see those regions. This type of prophylaxis really focuses on transmission prevention because it works in the upper respiratory tract. And as I said earlier, it's orthogonal to the immune system, fantastic for immunocompromised individuals, elderly people that we know that their waning is much faster than in the kind of like younger individuals, and the distribution is quite easy for something that you can self-administer. Okay, so how are we going to develop this siRNA cocktail? So we decided to take an approach in which we will not just try a few handful of siRNA molecules, but we will scan the entire genome of the virus. So if this is the viral genome, and it has about 30,000 places, we parse this genome into regions of roughly 22 nucleotides that are the targets of an RNAi trigger. And we look at all the regions that we can synthesize safely, that they are reasonable, that there is a little bit some bioinformatic prediction that they might be efficacious RNAi targets, and that they are safe, that they don't show any homology to the human genome, so we can target them without worrying that we're also targeting some native genes. Okay, so how are we going actually to simulate these like 16,000 RNAi triggers? If I'm going to order them from IDT one by one, this will take basically, it would cost like a fortune for a startup like us. So instead, we used Twist Biosciences. Instead of getting these RNAi molecules as siRNAs, we render them into these fragments over here, these DNA fragments, where we encoded the RNAi trigger as a short hairpin over here. And each one of these hairpins, the guide strand, targets a different region in the genome. There is a spacer, and after the spacer, there is the target region. This is basically 50 base pairs from the virus, including the target area that we want to get over here. And then with a few cloning steps, we can convert these fragments from Twist. We can convert them into this library of plasmids in which each insert has this RNAi trigger of interest. And the target site from the virus is in the 3' UTR of a Venus protein. So this way, if the RNAi trigger works fantastically well, it will go and repress the Venus expression. Now, the thing is that when you actually introduce this library into cells, it's a bit more complicated because sometimes when you introduce this reporter gene, sometimes it goes to the genome and integrates in an area where there is no good expression at all. So you might see blank cells and you think this is a fantastic RNAi trigger, but in fact, it's a quite lousy one. It just was very bad integration in some epigenetic region that doesn't allow the expression. So to overcome this issue, we developed this strategy in which we can turn on and off the RNAi machinery in the cells. And once we turn it on, so at the beginning it's off, we screen the cells only for cells that express Venus with very high expression, meaning that there is a good integration. Then we turn it on, and now we have a molecular knob that we can dial it up and down and kind of like find the best conditions to identify what are the most efficacious RNAi triggers. And we do it with basically flow cytometry, followed by high throughput sequencing that tells us where these RNAi triggers end up, in which condition. Now, as part of this library, we also introduced 1,000 controls of known highly efficacious RNAi triggers and very bad RNAi triggers from previous studies unrelated to SARS-CoV-2. And with these controls, this allows the training set to find kind of like the right knob, the right condition to separate between the bad ones and the good ones, and to find basically a precision recall curve over here. Now, as you can see, the curve maybe is not fantastic, but as a company, we are interested only in this kind of like region over here. High precision, meaning that I can find these hyperpotent siRNAs, but I don't really care about the recall because I don't need to find all of them, but just to find a handful of them. Okay, here are just like a slide to show that the screen is kind of like as a whole actually worked. We know from previous studies that shRNAs, highly efficacious shRNAs, don't like to have adenine in position 20. It doesn't work so well when they have adenine in position 20. So what you see over here, this is the screen results from the worst to the best RNAi triggers. And here is the probability that an shRNA trigger that got very good result will have adenine in position 20. And what you see is that there is a very good correlation. As we go to the best, the most efficacious RNAi triggers in our library, we simply don't see any adenine as expected. And this helps us to validate that the screen performs correctly. We did more layers of validation. I don't have time to show you, but just like I wanted to give you one example. The results of the screens are something like that. For each position along the genome, we have the prediction about whether this is a good RNAi target or not. We picked 10 positions, the first 5 just based on the high score, the other 5 based on conservation between SARS-CoV-2 and SARS, like the 2003 SARS, and their high score. So we had 10 RNAi triggers that then we moved into subsequent analysis and validation. Okay, so let me show you the results. First, we calculated the IC50 using a reporter assay for these 10 RNAi triggers. And what we found, quite fantastically, that for 5 triggers, we have IC50 of less than 20 picomolar. And that's amazing, because this is exactly what we wanted, very strong RNAi triggers. We also, this was in the reporter assay. Now we moved to a live virus experiment in a BSL-3 facility at Cambridge University with our collaborators. And you can see that for 5 out of these triggers, we see a reduction of over 95% of the virus. And this is by qPCR. By the way, the same qPCR that you do COVID tests to see whether you contracted it or not. And we also took the results from this screen, and in some conditions, we subjected the remaining virus and we measured with quantitative media the amount of live virus. So this is another approach to measure the repression. And you can see that for some of our siRNAs that we tested, we got a repression of tens to 100 folds of the virus. So it is working really, really well. To further improve this method, we also considered cocktails. So we took pairs of siRNAs from here, and we tried them together at the same concentration to the individual's one over here. Now, this is one in this figure is the repression that we get by the most individual efficacious siRNA, S3 over here. Now, once we start to put cocktails together of 2 siRNAs, we get extra repression because of synergistic effects. And of course, we like cocktails because they give resistance to escape mutants. Now, everything I showed you was done with the ancestral strain. Probably you ask, what about Delta? Does it work on Delta? And the answer is yes. We see that the same cocktails also work fantastically on Delta. You can see here the viral depletion. So basically higher, meaning more depletion of the virus based on qPCR. And you see that for some of the cocktails, we have very high depletion of the Delta strain. Okay, everything so far was done basically with cells. And now the question, of course, what about in vivo? So we did an initial in vivo experiment. We had 3 arms in this, what I'm showing you here is 3 arms of this experiment. We had a positive control. We took the FDA-authorized Lilly antibody, monoclonal antibody, and we gave to 6 hamsters this monoclonal antibody prophylaxically. And then we challenged them with the virus. For another 6 hamsters, we just gave them intranasal administration of siRNA that is not specific. And a day later, we challenged them with the virus. And we also took our best cocktail and subjected 6 hamsters to this cocktail. What we found was quite nicely, here is the positive control. It does protect these hamsters. Here is the negative control. It does not protect the hamsters. And you see here the treatment that goes really nicely in terms of weight loss protection with the FDA-authorized monoclonal antibody by Lilly. And the monoclonal antibody is systemic. We are just intranasally. And we did this experiment together with our collaborators at the NIH, and we're really excited about these results. So to summarize this part of my talk, I showed you a comprehensive screen to identify RNAi triggers. I showed you how we validated this screen technology. We found multiple hits that are highly effective, like showed RNAi triggers with very high potency, meaning low IC50. And we did many experiments in vitro to show that it actually represses the virus, and also presented some initial promising results from an in vivo study that we conducted a few weeks ago. So we are now repeating this in vivo study with different dosing, more conditions, and of course to replicate it and to see to optimize these results even further. And with that, I go to the second part of my talk, talking about a replicon system to simulate SARS-CoV-2. So here is kind of like one dirty truth about doing live virus experiment with SARS-CoV-2. It's really difficult, and it's difficult due to two things. The first one, you need a BSL-3 facility, and guess what? These facilities all over the world are quite in high demand. So it takes a lot of time to find a facility to develop the working relationships. Now, even if you find a facility, you still sometimes have issues getting new strains. So you work on one strain or the ancestral strain, you want to get alpha or delta or whatever. Usually these strains come with like a lot of like MTAs and lawyers that need to sign on them in order for you to avoid this, to actually get the strain. So basically, kind of like the take-home message from this slide, if you do live virus experiments, either you have to be in a BSL-3 facility or work with these really ugly MTAs, you need to take a shower at the end after that. And we wanted to avoid this situation and to find an alternative for that. Something that we can work in, not in a BSL-3 facility, and that we can very fast prototype new strains of virus. So the solution is a replicon system. What is a replicon system? It's basically an RNA molecule that retains nearly all the function of the virus. Not all, but almost all of them. It can, once you transfect cells, it produces the proteins of the virus, and it can replicate within the cell. So it has a very similar dynamic intracellularly to the virus, but it cannot go out and infect other cells. And therefore, it is a safe alternative because it cannot infect you. It's kind of like a virus that we chop certain functions that it cannot infect other cells. It's not a new idea. Replicon systems have been developed for hepatitis C, for the old SARS, and even for SARS-CoV-2. And I'm showing you one manuscript on SARS-CoV-2. But the problem with these kind of like academic efforts to develop these replicon systems for SARS-CoV-2 is that first, you cannot just go and buy them from anyone. They are usually not from scratch. So actually, you need to have first an access to the strain of interest, and then by a lot of PCR and stitching together, you get your replicon system. So it still has some strings attached. And they not always retain all the function, like nearly all the function of the virus. For instance, in this manuscript, they removed all the structural proteins of the virus besides the N protein. And if we have siRNA that targets these structural proteins, it will not work. So we cannot simulate the virus using this replicon. So we wanted to create this replicon system that will be based on de novo synthesis with Twist. We'll have a bioinformatic pipeline that we can just select a strain and very quickly get the strain of interest, send it to Twist as a FASTA file, and then get the replicon. We also wanted to add a reporter gene to save our qPCR costs and to have a design that will still maintain some of this, like as much as possible from the viral genome, and will be industrial ready. So this is the SARS-CoV-2 genome. There are three main N proteins on the outer side of the virus, spike that we all know. We also have the M, the membrane protein, the most abundant one, and we have the envelope protein. All of these basically get these viral particles that you see. So we knew that M is necessary to create these coronavirus particles, and we decided to totally remove this gene for safety. We also know from previous research that E is either necessary or at least highly important, because if you don't have the E, in some experiments, it highly attenuates the infectivity. So we removed E as well. And we know that if you remove E and M, basically you should not get any viral, the ability of the virus to form these particles and go out of the cell. As extra caution, we also, we know that spike is, everyone know that spike is essential as a receptor binding for SARS-CoV-2. So, and also that there is a furin cleavage site, that this is actually the main, like one of the main differences between SARS-CoV-2 and the rest of the coronaviruses that makes it much more infectious. And there was a beautiful Nature paper, so it's supposed to be 2021, not 2003 here in this Nature paper, that shows the importance of this furin cleavage site. So we changed the spike protein. We put an early stop codon so it cannot translate. And we also removed, for abundance of caution, also the furin cleavage site. So even if it does somehow translate and you also get the E and the M, somehow it will be very attenuated virus. And we also introduced a luciferase instead of the E and the M to have some sort of reporter gene. The design was approved by Cambridge University by a safety committee. The 30 kilobases of sequence of DNA was synthesized by Twist, and we in vitro transcribe it, and we conducted this experiment in BSL-2, again for the abundance of caution, although they should not infect you at all. It could be BSL-1. So safety first. This is by sequencing by Twist, no furin cleavage site of their construct, no E and M. I was a bit stressed that maybe someone will make a mistake and synthesize those, so we know that it doesn't exist there. We also did qPCR of the RNA of the virus, no E gene. Of course, we do see the RDRP because it's part of our design. Again, safety is fine. And the ultimate safety assay, we took cells, we transfected them with the replicon, we let the replicon replicate, and then we took the supernatant of the cells, infected naive cells, and wanted to see whether we see any viral infection over there. Here's the negative control of the qPCR, the positive control of the qPCR, and you see that basically there is 0 in our naive Vero E6 cells, meaning they cannot infect other cells, and this is the highest kind of like level of safety that you can imagine. So second, let's go to the next slide. Just to show that the replicon actually works, we transfected 3 types of cells, HEC293, hamster cells, and Vero E6 with growing levels of the replicon system. We also infected another for each cell type as a control, a luciferase mRNA, and we measured the luciferase expression of our replicon versus the level of luciferase with the mRNA in the other condition as a function of time after 24 hours, 48 hours, and 72 hours. And what you see that in nearly all conditions, the replicon signal versus the mRNA signal goes up, meaning that the replicon can survive and replicate, whereas mRNA, regular mRNA, goes down. So we are getting a replicon system that replicates, and this is exactly what we want. All these results really from the last week, I'm really excited about these results because it gives us a modular system that now we can accelerate our R&D efforts with SARS-CoV-2. We can de novo prototype any strain without dependency on patient's accessibility, lawyers, and so on. And there is a reporter gene that really allows us to get data quite quickly without qPCR. We established the results for BSL-2, and you can do basically experiments with this replicon. We established that you can do experiments up to 72 hours, and we are now evaluating it for our siRNAs to see that it's useful. So this is the acknowledgement slide here, the genome-wide screen, and the replicon team. I would like to highlight the involvement of Ahmed over here, a postdoc, basically used to be a postdoc at Cambridge University, joined the company quite recently, doing a fantastic job over the weekend to get you all these results, and our funding from the Bill and Melinda Gates Foundation. And if this is of an interest and you're in the Cambridge area in UK, you want to move there, please send us an email to hr@11tx.com. Thank you very much. And I would love to answer any questions.